name is Valentina Ortiz, and I'm a medical student at Universidad Pontificia Bolivariana. In this video, I'm going to talk to you about this article called Lack of K140 Immunoreactivity in Junction with Epidermolysis bullosa skin and keratinocytes associates with misfolded laminate epidermal growth factor-like MODIF2 in the beta-3 short arm. So I want to start off by giving a little introduction to the subject. Keratinocytes are highly specialized epithelial cells. They are the main cell type in the epidermis, which is the outermost layer of the skin. These cells originate in the stratum of the cell, where they can stay, where they act as stem cells, or they migrate upwards through the stratums of the epidermis. As they move upwards through the stratums, they start to get different functions, such as keratin production and protection of the organism as they separate it from its external environment. On the other hand, laminin is a crust-shaped glycoprotein, which consists of 3 different polypeptide chains. It is encoded in the genes LAMB3, LAMB3, and LAMB2. Laminin is assembled in the keratinocytes in the plasmatic reticulums and then secreted to the extracellular matrix, where it has a really important function as a major component of the epithelium basement membrane. The C-terminus part of the alpha-3 chain in the laminin has different binding sites or integrin receptors in the epithelial cell. This allows it to interact with the cell. As for the N-terminus of the gamma and the beta chains, connect to type 4 collagen and other basement membrane laminin. As a whole, laminin is really important for epithelial adhesion, and it's also involved in cell to extracellular matrix interactions. Now we're going to talk about K140, which is a monoclonal antibody directed to the beta-3 short arm of the laminin 332. It was found in this article that the region recognized by the K140 is the laminate epidermal growth factor-like MODIF2 of the beta-3 short arm. As shown on this graphic, where they compare 2 different beta-3 short arms of laminin, one has the ELE2 and one doesn't. The one that doesn't have the ELE2 doesn't have any reaction to the antibody K140, whereas the one that does have it reacts to the antibody, proving that the ELE2 in the beta-3 short arm is the region identified by the K140. Now, when we talk about immunoreaction, we're referring to the interaction between an antigen and an antibody. These two usually bind together by non-covalent bonds. Also, some immunochemistry techniques uses antigen-antibody reaction, its properties, or consequences to measure the presence of a certain molecule. Object. The main object of the study was through molecular characterization of a patient with junctional epidermolysis bullosa, recognize the sub-region identified by the monoclonal antibody K140. Methods and techniques. So the Western blot was one of the techniques performed in the study. This is an analytic method used to identify specific proteins in a given sample using antibodies. The 2 basic principles of the Western blot are protein electrophoresis and indirect ELISA. In a Western blot procedure, the proteins are first separated using gel electrophoresis. Then, they are transferred to a membrane, which is usually PVDF or nitrocellulose. Next, the sample protein is probed with primary antibodies, which are specific for the protein. And lastly, a secondary labeled antibody is used to further visualization. As the antibodies are specific, they only bind to the protein to be studied. So the only visible band on the results are for that specific protein. Also, the thickness of the band is directly proportional to the amount of protein present in the sample. In the study, they used Western blot to determine K140's reactivity to the patient's laminin in comparison to a control and another patient with LG domain mutation. They also used polyclonal antibodies to see if it would recognize the beta-3 chain in the patient. Another technique was immunofluorescence, which is based on the use of a specific antibody conjugated with a molecule of chlorochrome. In the technique, chlorochrome is used as a dye to tag the antibody. This molecule is characterized by its ability to absorb UV light but then emit visual light. In that way, we can easily visualize the reaction between the antigen and the antibody using a fluorescence microscope. In the direct immunofluorescence, the fluorescence present in the result is directly proportional to the amount of antigen present in the sample. Whereas for the indirect immunofluorescence, it's pretty much the opposite. In this study, this technique was used to evaluate the expression of laminin 332 in the patient's skin compared to a control. It was done using monoclonal antibodies K140, BM165, and GB3. Then we have RT-PCR, which is a technique used to determine the expression of genes in various tissues. It starts with an RNA strand as a mold, which could be messenger RNA or total RNA. The RNA then is transcribed into complementary DNA. This is done by an enzyme called reverse transcriptase. The complementary DNA then goes through the multiple normal PCR procedures to amplify it and make it easy for us to analyze. It could also be used as a template for qPCR, which quantifies the amount of RNA that has been transcribed in the sample. In the study, it was used to analyze the mutation consequences. Lastly, we have DNA sequencing, which is a technique that determines the order of the nucleotides that make the DNA molecule. It allows us to identify the information contained in a gene, and also it could highlight any changes in the DNA that may cause the disease. In this study, sequencing was used to identify any mutations in the patient and his parents' LAMB3 gene. Results. So the result number 1 is this immunofluorescence image over here, where we see that using monoclonal antibodies MB165 and GB3, we get weaker staining in the patient's skin compared to the control, while the staining was negative using K140. As for the second result, the first one shows us that the patient's keratinocytes show negative staining using K140. Using the GB3 monoclonal antibody, we see that the patient's keratinocytes show intense intracellular staining. In the second image, we see that the mutant protein and the ER-resident binding immunoglobulin protein show similar staining pattern. Finally, in the third image, we see that VIP expression is increased in patient keratinocytes. In the third result, we see the lack of reactivity of K140 to the patient's laminin, either in the media or cell lysates. Then we see high levels of unprocessed laminin in the patient lysates. The second image shows us that there was no reaction of K140 to the beta-3 chain, while the polyclonal antibody recognized it. Finally, in the fourth result, we see the DNA sequences of the patient and his parents. Here, we see that the mother has a mutation in the intron 10, which also has the patient. In the discussion section, I added 3 different authors' concepts. The first one was shown to be true because it was mentioned in the article that these kinds of mutation causes the mutant protein to stay in the endoplasmatic reticulum of the keratinocytes, and that reduces the amount of protein secreted to the extracellular matrix. The second concept was shown true as well because they did mention that the BIP protein was highly increased in these patients. And it was probably because of the protein accumulation in the endoplasmatic reticulum. As for the last concept, it wasn't true because it wasn't mentioned at all in the article.
As for my conclusions, the first one was that finding a way to induce laminin 332 that is retained in the endoplasmatic reticulum out of it. This may be a treatment aim for this kind of patients. The second one is that the antibody K140 is crucial in diagnosis of this subtype of junctional granulosis bullosa.